Hi everyone, this is a supplementary lecture on mutualisms. In this lecture, I'll expect you to know the material presented through slide 13. This slide explains the difference between the two major groups of mycorrhizal fungi. You won't be tested on any material after that slide. I'll cover that information, but it's just for your information only. So let's get started. As I mentioned, we covered a few of these slides, but I'm just going to recap them. Mutualisms are a diverse set of interactions between individuals of different species. They involve a wide array of species across a broad range of taxonomic groups. But what ties them together is that their interaction benefits both partners in the interaction. Generally, mutualisms are of three different types. There's the dispersive, defensive, and trophic mutualisms. Defensive mutualisms involve protection from predators, herbivores, or parasites, where one of the partners benefits from that defense and the other partner is provided with resources. We'll talk through one of those examples. A dispersive mutualism, as we mentioned, that's a mutualism where there is transport of gametes or zygotes between partners. And so in this example, we're going to talk about an example between plants and pollinators. The third major group are what are called trophic mutualisms, and trophic just refers to feeding relationships or nutritional relationships. These are mutualisms that involve the provision of essential nutrients between the interacting partners. So let's take a look at each one of these in turn. The yucca plant and its yucca moth pollinator are an interesting example of a dispersive mutualism. The plant depends on the moth to disperse its pollen and fertilize other plants, and the moth depends on the plant for a protected place with a ready food source to lay its eggs. Mutualisms vary in the degree of dependency between the species. In this example, the species involved depend completely on one another and cannot survive or reproduce without the other. The example of the yucca and the yucca moth mutualism is obligate because the yucca depends entirely on the moth to disperse its pollen, and the moth depends entirely on the yucca for its reproduction. Let's just take a look at how this works. The female yucca moth enters a yucca flower and makes a cut in the ovary with her ovipositor and deposits somewhere between 1 and 15 eggs. After the eggs are laid, the moth crawls to the top of the pistil of the flower and deposits a bit of pollen, which she had collected from another yucca individual or a number of other yucca individuals, and she deposits this pollen onto the plant's stigma. Of course, this guarantees that the flower is fertilized and that the moth's offspring will have developing seeds to feed on, because the eggs of the moth, once they hatch, begin to feed on the developing seeds. After the moth has laid her eggs, she scrapes some pollen off the anthers and adds it to the ball she's carrying in her mouth parts. She then flies to another flower and deposits the pollen. So, you might ask, after depositing her eggs, why does the moth spend extra time and energy to deposit the pollen she's carrying onto the stigma of the flower after she's laid her eggs when she could just fly off? Well, the behavior has been selected for because if the moth indeed doesn't deposit pollen onto the stigma, the yucca will abort the flower. Of course, this would mean that the seeds don't develop and the developing larvae would starve and die. So this has been selected for evolutionarily. Another example of an example of a different type of mutualism, that is, this is a defensive mutualism, and this is an example of ant-plant defenses. So there are quite a wide range of species of acacia that form mutualistic relationships with a group of ants in the genus Pseudomyrmex. And this is a defensive mutualism because the ants the ant populations that are colonizing the acacias actually vigorously defend the plant from herbivores. In return, the ants are provided a place to reproduce and shelter in the form of swollen thorns, and you can see that on the right-hand picture. However, the acacia species also provide food resources for the ants in the form of either Beltian bodies or what are called extrafloral nectaries. The Beltian bodies store protein, and so these are extra appendages off the leaflets of the acacia that are rich in nitrogen in the form of proteins in the ants, which of course the ants require. Ants also require carbohydrates, and the plant supplies these as well through extrafloral nectaries. These are extrafloral, meaning they're not in the flower; they're outside of the flower along the branches and axes of the leaves. And these nectaries provide nectar, and of course, nectars provide simple carbohydrates, which the ants can feed on. So the plant provides housing, basically, and a food resource for the Pseudomyrmex ants, and in return they vigorously defend their home and food resource from any herbivore that might come along. This particular this particular mutualism is therefore defensive, but it's also facultative because a number of different species of acacias and ants species can form these associations, but the plant actually, its survival doesn't depend upon the ants, but its reproduction is certainly reduced, which means that it's not obligate because the survival of the acacia doesn't depend exclusively on the ants or even a particular species of ant. And likewise, the ants can survive without the acacia, but both partners do better when they're in this mutualistic relationship. The third type of mutualism is referred to as a trophic mutualism. And trophic mutualisms involve feeding relationships between the partners, primarily in the form of transfers of nutrients, carbon and perhaps nitrogen, phosphorus, water in some cases as well. These mutualisms are basically all symbiotic. And by symbiotic, I mean that the two partners are essentially organically part of one another. An example of a symbiotic mutualism in human beings, of course, would be our gut bacteria and in association with us as the other partner. And the bacteria in our gut benefit from the environment provided by our gut system and food resources, and in return we receive the benefit of enhanced digestive capacity. In other organisms like lichens, plants and mycorrhizal fungi, corals and zooxanthellae, and plants and Rhizobium bacteria pictured here, all of these are examples of symbiotic relationships in which the two partners are organically part of one another. This differs from non-symbiotic mutualisms. The two that we just talked about before, dispersive and defensive mutualisms, are both non-symbiotic because the two partners actually have entirely separate physical existences. So let's take a look at these a few trophic mutualisms. First, lichens. Lichens are the two partners in the relationship in this symbiotic mutualism are a fungus living with either an alga or a cyanobacterium. In this mutualism, the alga or the cyanobacterium partner is autotrophic. So they're photosynthetic. They can fix carbon, which provides a carbon source for both species in the partnership. In return, the fungus provides basically a physical structure that is resistant to environmental extremes. As you probably know, algae and cyanobacteria aren't able to exist without, they exist in very moist environments, but yet lichens can occur on some of the most dry conditions, and this is because of the fungal partner providing basically a protective coating in its tissues around the alga or cyanobacterium. The fungus also provides access to nutrients, water and nutrients that are shared with the alga or cyanobacterium. Another example of a trophic mutualism is the symbiotic relationship between Rhizobium bacteria. This is a genus of bacteria that colonizes the roots of plants in the legume family, in other words, the pea family. This image here shows a close-up of the root system of a leguminous plant, a plant in the pea family again, and those nodules that the red arrows are pointing to are nodules that are formed out of plant tissue that house the bacteria inside. And this is important because the Rhizobium bacteria, which are able to fix nitrogen out of from gaseous form of nitrogen in the atmosphere. You may or may not know that the form of nitrogen in the atmosphere is N2. It has a triple covalent bond and requires, but that form of nitrogen gas is not available to plants as a nutrient, and so the Rhizobium bacteria are capable of breaking that triple covalent bond in N2 and converting it into ammonium, which it's an ion that is available for uptake and incorporation and assimilation into proteins and other molecules in plant tissues. Rhizobium require an anaerobic environment in order to conduct this chemical reaction, this fixation reaction of nitrogen. The nodules provide that. So within these nodules, the environment is anaerobic, that is, there is no oxygen. And so this provides basically the environmental conditions required by the Rhizobium to effectively fix nitrogen gas from the atmosphere and convert it into ammonium that can be used by the Rhizobium bacteria, but also is shared with the plant partner. So the plant benefits because it gets access to that nitrogen, and the Rhizobium bacteria benefit because they are provided with a structure within which they can maintain populations in this anaerobic environment. And the plant provides them with water and other nutrients such as phosphorus and so forth. This makes leguminous plants rather competitive in nutrient-poor, particularly nitrogen-poor environments, and often, and you may be aware of this as well, leguminous plants are often used in crop rotations to add nitrogen to the soil because they are very good at accessing nitrogen through this trophic mutualism, and the plants are then turned into the ground, and when they decompose, they release all that nitrogen back into the soil. Another example of a trophic mutualism is the mutualism between coral polyps, corals, that is, which are colonial, and zooxanthellae, which the coral polyps house. Zooxanthellae are dinoflagellates, which are photosynthetic algae, basically. And so most zooxanthellae are autotrophs and provide the host, in this case coral polyps, with energy in the form of reduced carbon compounds derived from photosynthesis. So there are many, many different species of corals, and these corals have a number of different species of zooxanthellae associated with them. So this is a facultative type of mutualism. And zooxanthellae can actually provide up to 90% of a coral's energy requirements. Here's a picture, a closer picture of coral polyps, and then the zooxanthellae inside those the tentacles there. Those small green dots are the zooxanthellae. So the zooxanthellae can provide up to 90% of a coral's energy requirements, it turns out. The rest of the energy is acquired from small fish and plankton and so forth, using various stinging cells called nematocysts on the coral polyp's tentacles. In return, the coral provides the zooxanthellae with protection, shelter inside of their tissues, and a constant supply of carbon dioxide that's required for photosynthesis. So the carbon dioxide is dissolved in the water, and it diffuses into the coral polyps, where that carbon dioxide is provided to the zooxanthellae to fix that carbon into carbohydrates through photosynthesis. So the zooxanthellate corals require sunlight, of course, because of this interaction, and grow best in clear, shallow water that's typically at depths shallower than about a couple hundred feet or so that they can get access to the light that they require in order to conduct photosynthesis. So again, this is a trophic mutualism because of the transfer of compounds between nutrient elements between the partners. The zooxanthellae fix the carbon and provide that to the coral polyps, and in return the coral polyps provide shelter, but also access to water and other nutrients that the zooxanthellae need, such as nitrogen and phosphorus, for example. Zooxanthellae are major contributors to the physical structure of coral reefs. They're tremendously diverse, and coral reefs develop in warm tropical and subtropical waters. An example, of course, is the enormous Great Barrier Reef off the coast of Queensland in Australia. So they are abundant in tropical latitudes because they require this warmer water. They require access to sufficient light, so they don't tend to be very deep. And as a result, they also are intolerant of turbidity, which would, prevent the sun from, sun's energy from reaching the coral polyps. They also are susceptible to changes in pH and so forth. And you might recall when we talked about one of our very first lectures, when we talked about climate change and increasing carbon dioxide in the atmosphere, a lot of that carbon dioxide gets dissolved in the oceans. And as a consequence of that, increased carbon dioxide in the ocean lowers the ocean pH. And corals are very susceptible to what's called coral bleaching. In other words, basically the corals eject their zooxanthellae partners when the pH gets too low. And this can cause major die-offs of corals. And this is something that we've been seeing worldwide due to acidified waters in tropical locations. So the last group of organisms that I want to talk about, last mutualism that is a trophic mutualism, are mycorrhizae. And so what are mycorrhizae? Well, mycorrhizae are a group of fungi that associate with plants. And the term mycorrhizae comes from myco, which means fungus, and rhiza, which means root. So these fungi colonize plant roots, and again, this is a trophic mutualism, so there's an exchange of nutrients between the two partners. So let's take a closer look at this mutualism. In the plant mycorrhizal mutualism, the two partners, of course, are the plants and the mycorrhizal fungus. The fungus benefits by receiving carbon from the plant. So when the plant fixes carbon through photosynthesis in its leaves, it translocates that carbon down into its root system. And because the fungus is colonizing the plant through its single cell filaments, the plant translocates carbon to the fungus, and so the fungus benefits by receiving a portion of the carbon that is fixed from the plant. In return, the plant benefits by receiving nutrients and water from the fungus. So, as I mentioned, the fungus, which is kind of illustrated here in this fruiting body. If you look at the base of the fruiting body, you can see these filaments extending from that fruiting body into the soil surroundings. Those filaments, those individual hyphae, when they contact a plant root, they can colonize the plant root, and so this is where that organic, coming together of the plant partner and the fungal partner happens, and this is where nutrient exchange happens. So the fungus, these fungi are very good at acquiring nutrients, both from the surrounding soil in terms of inorganic nutrients that are available, but many of these fungi are also very good decomposers, so they can acquire nutrients from litter, organic forms of nutrients, and also provide those to plants. The fungi are also very good at accessing water, and so they can provide water as well to the plant partner. So let's take a look. We'll talk a little bit more about this, but let's first take a look at some of the main functions of mycorrhizal fungi. So obviously, they improve nutrient and water uptake for their plant hosts. But they can also increase the plant host's tolerance to drought because, again, they're very good at accessing water from very small pore spaces. Soils have a variety of different pore spaces, and because the fungal hyphae are so small, they can access these very, very small pore spaces where plant roots are much larger in diameter and don't have access to those very small pore spaces. Mycorrhizae can also increase host tolerance to saline environments, even to heavy metals. A number of different species of Ekta mycorrhizae have been demonstrated to sequester heavy metals in their tissues, thereby protecting plants from their toxic effects. You might recall when we talked about serpentine soils, how these are soils common in California that have high heavy metal contents, and so some of these mycorrhizal fungi are playing a role in sequestering heavy metals out of soils such as serpentine and protecting the plants from their toxic effects. Evidence also suggests that these fungi can protect plants against pathogens, soil pathogens, as well. They can also alter competitive interactions. Plants with mycorrhizae have greater competitive ability because they have greater access to water and nutrient resources compared to some plant species which may not have mycorrhizal associations. They also can stabilize the soil as well. This stabilization of the soil results from the fact that the hyphae of mycorrhizal fungi can form quite dense networks of hyphae, sometimes called mycelium, and this will interconnect the soil particles and organic matter and so forth in a soil and result in soils being more stable and therefore less susceptible to nutrient loss through erosion and so forth. There are two major groups of mycorrhizae, endomycorrhizae and ectomycorrhizae. These two broad groupings differ taxonomically, evolutionarily, and also morphologically, and in a number of other respects. They differ, first of all, the endomycorrhizal fungi are referred to as endomycorrhizae, endo meaning within, because the hyphae, the filamentous hyphae of these fungi actually penetrate the plant's cell wall. And when they do, they invaginate the plasma membrane of the plant cell in the roots. And this juxtaposition of the fungal hyphae and against the plasma membrane within the plant cell is where nutrient exchange happens. This differs in ectomycorrhizae, ecto meaning outside of, because the fungi, the hyphae of ectomycorrhizae do not penetrate the cell wall. The fungi in the hyphae instead grow in between the cortical cells in the root, and it's in this part of the root, in the cortical cells, where the hyphae are adjacent and in contact with the cortical cells of the root. This is where nutrient exchange happens in the ectomycorrhizae. The endomycorrhizae form arbuscules, and vesicles, and we'll take a look at some of those in the subsequent slides. So I won't go into much detail there. But they differ on a microscopic level at the cellular level, in that they form arbuscules. And arbuscules are arb, meaning tree. They form these tree-like structures when they invaginate the plasma membrane. And again, this is where it's in these arbuscules. Why can't I say that? Arbuscules where the nutrient exchange occurs. Vesicles are sort of storage structures. By contrast, in the ectomycorrhizae, when the hyphae penetrate the roots and grow in between the cortical cells, they form what's called a Hartig net, or a kind of a network of hyphae in between the cortical cells. They also form a sheath, or a kind of a blanket, if you will, of hyphae around the entire root. And so in some subsequent slides, you'll see that. The endomycorrhizae do not have, they virtually have no external structures, and they do not produce sporocarps, that is, fruiting bodies. So you don't see like mushrooms, growing, any mushrooms you see growing on the forest floor or what have you. Those would not be endomycorrhizae, because there are no really known sexual reproductive structures in the form of sporocarps that have been identified. In comparison, the ectomycorrhizae form pretty extensive hyphal networks. So I mentioned those in terms of soil stabilization. So they form a lot of external hyphae, and these hyphae penetrate the soil volume and, are extracting nutrients and water and so forth. Ectomycorrhizae do produce sporocarps. These are the reproductive structures. So, in other words, mushrooms. So if you see a mushroom on the forest floor, it may in fact be an ectomycorrhizal species. Not all fruiting bodies or sporocarps are ectomycorrhizae. Of course, many of them are decomposers, and they're not mycorrhizal at all. So you can't be sure, but ectomycorrhizae are known to produce visible fruiting bodies. Endomycorrhizae occur on most vascular plants. It's estimated that they are on somewhere upwards of 90% of all flowering plants. So they're very, very broadly distributed across an enormous number of plant species. The ectomycorrhizae occur on very specific taxa. In particular, they tend to occur on species of conifers, so pine, spruce, fir, and so forth. They also occur on oak species, eucalyptus, and some other specific taxa. Although they don't occur on as many taxa, they're actually very widespread because those taxa that they do occur on are quite widespread worldwide. It's estimated that there are between 5,000 and 6,000 species of fungi that form ectomycorrhizas, and around 4,500 or so of these are what are called epigeous, and that means that they have above ground fruiting bodies. Up to a quarter of them or so produce below ground fruiting bodies. These are called hypogeous fungi. An example of a hypogeous fungus would be, or of a sporocarp or fruiting body of one, that is, would be like truffles. So if you have ever had truffles, then you know what I'm not talking about the chocolate kind either. These are fungal fruiting bodies, and they're considered delicacies in a number of different cuisines. So at this point, the remaining slides will go into some greater detail about endo and ectomycorrhizae, but you will not be tested on this material. From this slide forward, it's for your information only. So let's take a closer look at arbuscular mycorrhizal fungi, or AM fungi. These are endomycorrhizal species. They form host associations on they're on the roots of about 80% of plant species, and that's equivalent to about 2/3 of all land plants. They're around 90% of if you consider just vascular plants, they occur on about 90% of all vascular plants, including many crop plants. So these are very, very important species. They have been around for a long time evolutionarily. We have fossil evidence that these fungi have been around since about 400 million years ago, before plants actually colonized, or about the time that plants were colonizing land. So these species probably played a very critical role in the plant's ability to colonize land at the time. Morphologically, they produce, as I mentioned, arbuscules and vesicles. Here I am stumbling over that again. An arbuscule is shown below. There are those black and white images, and you can see why they're called arbuscules, because they do look sort of arbuscular, like trees. This, the arbuscule, is a structure that forms when the fungal hyphae, after they've penetrated the cell wall, they'll invaginate the plasma membrane and form these very complex tree-like structures, again, arbuscules. And this is where nutrient exchange occurs, because this is the contact point between fungal tissue and the plant cell. All right, I'll just I'll continue on and talk about the structures a little bit more. This is a diagrammatic view of arbuscular mycorrhizal fungi. On the left, you can see sort of a cross-section of a root, and that cross-section includes, in this three-dimensional image in the middle, the epidermis, of course, and that's where you get the root hairs that extend off of the plant root in order for nutrient uptake. The purple structures are fungal hyphae. So these are single-cell filaments or hyphae that are, you can see, are penetrating the plant root. So the hyphae are growing in between the epidermal cells and penetrating the root there. You can also see that there are arbuscules as well. So the cortical cell, those are cortical cells there. And those arbuscules are where the hyphae has penetrated the plant cell wall and invaginated the plasma membrane. On the right, you can see a stained specimen showing arbuscules as well in purple. Okay, and the cortical cells are labeled there as well. So these arbuscules, again, are where the nutrient exchange occurs. In particular, arbuscular mycorrhizal fungi are very efficient scavengers of phosphorus. So they can also access other nutrients, but they tend to be somewhat more specialized in accessing phosphorus from soils. So this probably happens through a variety of different mechanisms, due in large part probably to just simply the increased surface area of the many small hyphae that extend into the soil. But these fungi also produce enzymes called acid phosphatases. And so these phosphatases are able to break down organic forms of organic molecules that have phosphorus in them. So these phosphatases are secreted into the surrounding soil by the colonizing arbuscular mycorrhizal fungus, and they release, these enzymes are released, and then this liberates phosphorus from that are, it's bound in organic molecules. Whatever is not used by the fungus can be transferred to the plant host, and so the plant host benefits from increased access to this phosphorus. Different species of mycorrhizae vary in their effectiveness for taking up and transferring phosphorus to the host. So depending upon what particular species is associated with a plant, that will influence the ability of the benefit that the plant receives of increased phosphorus uptake. There are many studies that have very clearly shown that plants colonized with arbuscular mycorrhizal fungi can significantly increase plant growth. So this is just simply an illustration of that plants on the right were grown with mycorrhizae, plants on the left without, for two different plant species. So hibiscus on the left, lemon trees on the right, and the individuals on the right are those that have that are colonized by mycorrhizal fungi. So this has been demonstrated in quite a wide range of plant species, and with different species of arbuscular mycorrhizal fungi as well. We also know that arbuscular mycorrhizal diversity can alter the nutrient uptake and also biomass in a grassland community. So researchers working with arbuscular mycorrhizal fungi have investigated how, in this case, a grassland ecosystem of 15 different species would respond to different numbers of fungal species in the soil, 15 different species of plants, that is. When they looked at the total amount of phosphorus that the plants took up from the soil, they found that plants living in soils that contained more fungal species took up a greater amount of phosphorus. And this is shown in panel A. Okay. They also quantified the total biomass of roots in the soil, the plant roots that is, and shoots, plant shoots above the soil. And what they found was that a higher number of fungal species in the soil caused a substantial increase in the biomass of roots and shoots of the plants. And this can be seen in figures B and C in the panels here. So, as is illustrated in this example, mutualisms not only affect individual species of plants, but they can also have large effects on plant communities through influencing these kinds of ecosystem processes, that is, nutrient uptake capacities of plants in plant communities. So now let's turn to ectomycorrhizal fungi. So these are some images of what are called mycorrhizal root tips. So the roots, plant roots, when they're colonized by ectomycorrhizal fungi, form fairly visible and distinctive morphological structures. And some of these are shown here in these pictures. So on the top left image, You see the root, the uncolonized plant root, is this sort of rusty red color. The root that is colonized is black, and you can, if you take a close look, you can see that there is root tissue that's completely enveloped in fungal tissue. The fungal tissue here is appearing black, and the roots are kind of clubby and pudgy, if you want to call them that, and they have this interesting sort of branching pattern as well. In all of the four pictures, you can see these sort of clubby root structures. In the top right, the plant root is kind of, again, this rusty brown color, and the colonized roots are kind of cottony looking, kind of whitish cottony. And the lower images as well, you can see these very dramatically modified kind of stubby roots, and those are segments of roots that are colonized by ectomycorrhizal fungi. Ectomycorrhizal fungi, as I mentioned, occur on particular taxa like pine and fir, spruce, poplar, willow, beech, oak, eucalyptus, all sorts of species. They don't occur on as many species of plants as endomycorrhizae do, but those species that they do occur on are very, very widespread, particularly in temperate and boreal latitudes. I talked to you a little bit about the morphology, or I'm going to, and I'll talk about that a little further in subsequent slides. They colonize plant roots again by, they penetrate the plant root, but they don't penetrate the cell wall. They grow in between the cortical cells and the roots, and we'll take a quick look at that as well. Their reproductive structures are fruiting sporocarps that fruit above ground. And as I mentioned again, they occur on maybe 5,000 to 6,000 different plant species. Some of them are very specific to certain plant species, but most are pretty broad generalists. Okay, so mycorrhizal fungi are a pretty widespread trophic mutualism. This just kind of gives you a cutaway image of what it might look like below ground. This is an example for ectomycorrhizae, since this is the group that we're talking about here. So, taking a closer look below ground, this image of a little seedling, obviously, is illustrating the above ground portion, but what's going on below ground? In particular, what's going on with respect to mycorrhizal colonization, and in particular ectomycorrhizal colonization. So this image here, actually, this is an image I grabbed off the cover of a book called Mycorrhizal Symbiosis, and this, I think, is just a really very impressive image, because what it shows is that there's an enormous amount of colonization going on below ground. And in particular, if you look at this image, what you'll see is that you have a seedling in the center. There are a couple seedlings on either side of that central seedling, but let's just focus on that central one. What you can notice is that the plant root is the main, is this rusty colored structure, and you can see that there's sort of a main taproot going straight down, and then two lateral roots. But that's it. I mean, that is the entire root structure for this plant. But what you should also notice is that connected to that very simple root system of this seedling is an enormous amount of fungal hyphae in white, and you can see that sort of reticulate that reticulate structure that is all around the roots and penetrating into the surrounding soil. What you should also notice here is that there are certain points of contact or colonization that are happening. These are mycorrhizal roots. So this is the point of contact with the plant and where the mycorrhiza is colonizing the plant. In other words, penetrating the epidermal cells and growing its hyphae into the root and in between the cortical cells. And of course, again, this is where nutrient exchange is going on. So the fungal hyphae actually provided an enormous extension of the plant's root system, and therefore the ability of the plant to access water and nutrients and so forth that it otherwise would not have access to. So taking a closer look, the diagram on the left shows one of those clubby roots that I had showed you in the first slide, but close enough to where you can actually see the fungal hyphae that's enveloping the root. And this is called a sheath or a mantle. Both of those refer to the same thing, just this covering that occurs around and envelops the root, and it's basically fungal hyphae. In the center diagram, you can see, just like in the previous slide showing the endomycorrhizae, we have the epidermal cells, we have the cortical cells just beneath that, and in purple is the fungal hyphae. And so you can see that the fungal hyphae is enveloping the root in the form of that mantle or sheath, and then individual hyphae grow in between the epidermal cells, and so they're penetrating the root that way, and they're growing in between the cortical cells. So notice, in contrast to the arbuscular mycorrhizal fungi, that the hyphal filaments of the Ekta mycorrhizae do not penetrate the cell wall. They grow in between the cells in this case, and they form a network. And this network of hyphae growing in between all of these cortical cells in the plant root is called a Hartig net. Okay, and you can see in that SEM diagram on the right, the hyphae are basically shown kind of looking like a skeleton around the cell. So the cells have been, I think, enzymatically removed, and the fungal hyphae remains. So this is where nutrient exchange occurs. Okay, kind of in a stepwise fashion, kind of zeroing in. On the upper left diagram, again, this is an ectomycorrhizal root, or root tip sometimes they're called. So those clubby, kind of cream-colored clubby roots, those are roots that are colonized by ectomycorrhizal fungi. That rusty-colored central root is uncolonized. If we took a cross-section of one of those ectomycorrhizal root tips, the roots that are colonized, you would see that there's a sheath or mantle surrounding it. The arrow is pointing to that. And then in between all of those cells inside of that sheath, underneath the epidermis, those are cortical cells. And so the fungal hyphae are growing in between those cells, and that is what forms the Hartig net. The lower right diagram just zooms in a little bit closer to show you a single cortical root cell and the fungal hyphae that is essentially enveloping that cell. Okay, and so that's just a close-up of a couple different cells, cortical cells, and the fungal hyphae forming that Hartig net. This, of course, and that big dark arrow going both ways, is where nutrient exchange occurs. So the plant is acquiring water, nutrients in the form of nitrogen and phosphorus, and in return the fungi is receiving carbon from the plant in the form of carbohydrates that were, of course, originally fixed via photosynthesis in the leaves and translocated down into the root system. On that lower left image is just showing the arrow is pointing to the fungal hyphae that is growing, kind of penetrating between the epidermal cells and beginning to grow around the cortical cells. And so that in this image looks sort of kind of like this brain-like structure. That's all fungal hyphae growing in between those epidermal cells and moving into the cortical area of the plant root, where the Hartig net will continue to form and where nutrient exchange will happen. So over the last number of years, over the last maybe 10 to 15 or so years, researchers have began to realize that individual plants, while they supported different mycorrhizal fungi on their root systems, they discovered that plants were actually connected below ground by fungal hyphae. And these are what are called common mycorrhizal networks. So common mycorrhizal networks are essentially networks of fungal hyphae that are connecting plants below ground. Okay, so that may be plants of the same species, so two individuals of the same species may be interconnected by the same fungal species or different fungal species. Individuals of different species as well are also connected below ground. And so research has shown definitively that nutrients are moving between individuals of the same plant via these mycorrhizal networks. So, for example, a study done by Suzanne Simard a number of years ago, this was research published in Nature, demonstrated clearly that carbon that was fixed in one plant ended up in a neighboring seedling of the same species. And this was conducted using stable isotopes, where individual nutrients can be tracked from one location to another. And so they provided carbon dioxide to a mature tree, and that was labeled as carbon-13, and that carbon-13 ended up being detected in a seedling in the forest understory. Additional research has shown that nutrients can not only be transferred from one individual to another of the same species using similar kinds of isotopic studies, but to individuals of different species. And so nutrients are moving around below ground between individual species, and this has really dramatically changed our understanding of the dynamics of plant communities. These mycorrhizal networks are extensive and very common, and have other functions as well. Some recent work has indicated that even anti-herbivore defense compounds are moving between individuals of the same species. When one plant is damaged, a signal, it can be transmitted from one individual to another of the same species that there's an herbivore outbreak, and that it can begin to upregulate anti-herbivore defense compounds. So this is just really fascinating and really interesting work that's being done that is really illuminating our understanding of how plants in communities are really quite connected to one another. Water has also been demonstrated to move through common mycorrhizal networks between plants, and so it could be that plants are more resistant to drought when they're connected into these networks as well. All right, well, that concludes the lecture on mycorrhizal, on mutualisms. And again, just I'm only going to hold you responsible for the information I presented up through slide 13. Anything after that is just for your information only.